Good morning and hello everybody. I'm going to have a review on obesity today, in which we are going to learn about regulation of food intake. I'm Dr. Shivaji Chalak, Assistant Professor from Department of Physiology, JN Medical College, Savangi, Wardha. Now the contents of today's presentation would be introduction, history, definition and indices of the obesity, prevalence of obesity, etiology of obesity, the hypothalamic centers and their role, and in the last we are going to see about physiological regulation of energy balance and food intake. As you all know, obesity can be defined as an excess of body fat or adipose tissue, simply, and a surrogate marker of body fat content is a body mass index. BMI can be calculated by weight in kilograms per height in meters square, and clinically the BMI is 25 to 29.9 kilograms per meter square. It is called as overweight, and anything beyond 30 kgs per meter square is called as obese. Now, with this introduction, I would like to tell you that obesity is a growing problem. The prevalence is increasing in the developing countries and most of the developed countries also, like US. It is not only the problem of adults, but also it is the problem of children. According to the WHO source, 1995, 2008 and 2004, WHO has given some principal cutoff points. Here we can call it overweight if it is more than or equal to 25. The BMI, pre-obese can be labeled in the category of 25 to 29.9 BMI. The obese more than 30. In obese also we have got sub-categories like class 1, class 2, class 3. Accordingly, they are divided into 30 to 34.9, 35 to 39.9, and more than 40 is class 3 obesity. Now in humans, the presence of nutritional abundance and a sedentary lifestyle and genetic influences increase the adipose energy stores and produce adverse health consequences. Now this is all because of the sedentary life and the faulty techniques of food eating, like junk foods and lack of regular physical activities and exercises. Now this obesity can alternatively be determined on the basis of percentage of total body fat. In males, 25% or more total body fat, we can label them as obese, and in females, 35% or more total body fat, we can label them as obese. Coming to some historical aspects, there is the earliest evidence of obesity. It was detected in Venus figurines and statues of obese females roughly around 24,000 BC to 22,000 BC, and was discovered by archaeologist Joseph in 1908. So since then, this obesity is detected, and we can call it as a problem of ancient era also. It was in 1951 when Professor B.K. Anand from All India Institute of Medical Sciences, Delhi, along with Professor J.R. Brobeck, both of them have done a lot of work on hypothalamic feeding center and satiety center. This experimentation was done on rats after stereotactic experimental techniques. Professor B.K. Anand and Brobeck, they destroyed the lateral part of hypothalamus, and they found that the rat was having no interest in eating. Later on, they destroyed the ventromedial nucleus of the hypothalamus and found that the rat was voraciously eating. And according to these destructions of these nuclei, they could come to a point that there are feeding centers and satiety centers which are situated in the hypothalamus. This was a great contribution as far as the regulation of food intake was concerned, and Professor Anand and Brobeck was on verge to get a Nobel Prize, but due to some reasons was not given. In 1996, the BMI was first published. Over the years, it seems to worsen despite of growing awareness, combating techniques, and now it has been called as an epidemic of developed countries. Look at the prevalence of obesity. There is a global prevalence. We can say more than 1 billion adults are overweight and at least 300 millions are obese. Prevalence is increasing in the United States and other developed countries also. Adults and children are the equal sufferers. If you look at the figures, in US, 64% adults are overweight and 33% are obese. The 16% are obese children. In India, 15% of population, according to NFHS data, with highest in Punjab, having 30.3% amongst male and 37.5% amongst female. So according to these figures, we can just see the grievousness of this problem, and the increasing prevalence is becoming epidemic nowadays, not only in developing countries but also in developed countries like United States. We'll come to the etiology of obesity. The obesity results from energy intake in excess of energy expenditure. So whenever there is imbalance between energy intake and energy expenditure, leads to obesity. And we all know the reasons is sedentary life, lack of physical activity, eating voraciously, junk foods, and faulty eating habits and faulty timings of eating. There are two types of obesities: hyperplastic obesity and hypertrophic obesity. In children, it's most commonly seen is hyperplastic obesity, in which the number of the adipose tissue increases as compared to the size of the adipose tissue. And in adults, we can see not only the increase in number of adipose tissues are there, but also the size of the adipose tissue also increases. So we can call in adults the obesity is not only hyperplastic but hypertrophic. There are lifestyle and environmental factors. The rapid increase in the prevalence of obesity in past 20 to 30 years emphasizes its role, as genetic changes would not have occurred so rapidly. So it's the lifestyle and the environmental factors. Sedentary lifestyle, as I am telling you, is a major cause of obesity. Cultural factors like availability and composition of food, that is the quality of food, is very important, and level of physical activity. This is deteriorating day by day. The physical activity is almost nil, and the quality of food, which include junk foods and fats, cheese, and the fatty contents, these are all contributory factors. We can call it as cultural factors. Psychological factors like stressful situations. Many people gain their weight during stress. Seems that eating has become a means of releasing tension. So the psychotic patient of depressions, they are stressful and they are involved in eating more. Next factor is childhood overnutrition. The rate of formation of new fat cells is especially rapid in the first few years of life. The rate is more in the fat storage and the number of fat cells. Hence, the overnutrition in the children in infancy can lead to lifetime obesity. There are some neurogenic abnormalities like lesions in the ventromedial nucleus of the hypothalamus that leads to hyperphagia and obesity. And there are some certain hypophyseal tumors encroaching upon the hypothalamus that leads to obesity. Although these two factors are little bit rare, and such hypothalamic damage is very rare cause of obesity. However, functional organization of hypothalamus and other feeding centers in obese do differ from non-obese. There is a set point in the hypothalamus. Now, when an obese person is asked to lose the weight by doing strict dietary restrictions, after losing the weight, it has been found that the hunger appetite, the hunger is more increased vigorously as compared to the non-obese person. So this determines that there is a set point in the hypothalamus, and this set point is at a higher level in obese persons as compared to the non-obese persons, and therefore they are involved in eating more. There are certain genetic factors, like obesity runs in families. About 20 to 25% are caused by genetic causes. Some mutations in the MCR receptors and in leptin genes and leptin receptors. Specific genetic syndromes like OB gene mutation is genetically seen in obese mice was a major breakthrough. Now we can see some obesity genes in humans and in mice. OB gene, it's a leptin gene. Now this causes, the mutation causes deficiency preventing satiety signals. So signals from the satiety center are prevented. It is present in human as well as in mice. Leptin R receptors, again the mutation of these receptors causes deficiency preventing the satiety signals. Now in mutations of POMC, the pro-opiomelanocortin neurons, there is mutation which leads to synthesis of decreased MSH, that is melanocyte stimulating hormone. MCR4 receptors, these are receptors of MSH, they prevent the satiety signals. And finally, agouti related peptides, these are neuropeptides which decrease the signals from MCR4 receptors. These are certain mutations of genes of leptin as well as the other neurons of the hypothalamus. Coming to the physiological regulation of food intake. Now as we know, for a stable weight, there should be total energy expenditure which should be equal to total energy intake. And whenever there is an imbalance between energy expenditure and energy intake, the person becomes obese. When a person is overfed and energy intake exceeds persistently the expenditure, most of the excess of energy is stored as fat in the adipose tissue and body weight increases. Now this stored fat can be stored either subcutaneously as adipose tissues or adipocytes in the adipocytes present in the subcutaneous tissue one, or in the visceral organs like liver and other organs. It is the visceral obesity which is more fatal as compared to the subcutaneous obesity. We can see for each 9.3 calories of excess of energy, 1 gram of fat is deposited in the subcutaneous adipose tissues. There are the neural regulators, neural centers are present which regulate the food intake, and hypothalamus is the important major site for neural centers. Professor B.K. Anand and Brobeck has discovered, they have together discovered the satiety and the feeding centers. Now the lateral nuclei of the hypothalamus, it acts as a feeding center. It operates by exciting the motor drive to search for food, and it is chronically active and inhibited by satiety center. Ventromedial nuclei, they act as satiety center. Primary center that it controls, it acts as a primary center and controls the food intake, and it functions by inhibiting the feeding center. There are other centers, other nuclei also, in addition to the ventromedial and ventrolateral nuclei, which acts as a satiety and feeding centers. We have other nuclei like paraventricular nuclei and dorsomedial nuclei. The lesions of paraventricular nuclei results in excessive eating, and the lesions of dorsomedial nuclei results in decreased eating. We have arcuate nuclei in the hypothalamus, which acts as a site where multiple hormones from GI tract, they converge and act upon these arcuate nuclei, and the adipocytes converge to regulate the food intake. The hypothalamus receives signals, one from GI tract, because of the distension of GI tract after eating food. 2. The chemical signals from nutrients in the blood. An important signal is the plasma glucose level. We have the different GI hormones like insulin, cholecystokinin, ghrelin, and leptin from the adipose tissues. We have hormones from the adipose tissues like leptin, signals from the cerebral cortex like sight, smell, and taste. In the figure we can see various hormones like GI hormones like insulin, CCK, polypeptides, leptin from the adipose tissue. They all have the inhibitory effect on the hypothalamus, on the feeding center of the hypothalamus, that is situated in the ventrolateral aspect of the hypothalamus. There is only one hormone, that is ghrelin, which is secreted from the oxantic cells of the stomach, and this ghrelin has an stimulatory effect, on the feeding center of the hypothalamus. Rest all the hormones, they have an inhibitory effect on the feeding center. So there is only one hormone, ghrelin, which is acting against a number of hormones like leptin, insulin, CCK, and neuropeptides. There are certain orexigenic and anorexigenic substances. The orexigenic substances include the neuropeptide Y, which acts as a neurotransmitter, the agouti-related peptides, orexins A and B, endorphins, cortisols, and ghrelin. We have another category, the anorexigenic substances, which includes alpha MSH, that is melanocyte stimulating hormone, leptin, insulin, cholecystokinin, POMC, and CART, peptide YY. The POMC and CART is pro-opiomelanocorticoids and caffeine amphetamine regulated transcripts. Now this figure diagram shows the nuclei of hypothalamus, the arcuate nuclei, and the paraventricular nuclei. Here I would like to tell you there are two types of neurons. One group of neuron acts as a orexigenic neurons, and another one acts as anorexigenic neurons. The POMC and CART, when they are stimulated, they release alpha MSH, which acts upon MCR4 receptors in the paraventricular nucleus of the hypothalamus, and through this nucleus they stimulate the feeding center, which is as present on the lateral aspect of the hypothalamus and is responsible for increasing the food intake. This is one pathway by which decreasing the food intake. This is one pathway by which they decrease the food intake, and the other pathway is the projections are projected to the nucleus of the tractus solitarius in the medulla oblongata, and through this nucleus of the tractus solitarius, the vasomotor center is stimulated by increasing the sympathetic activity and increasing the energy expenditure. To summarize, the stimulation of POMC and CART neurons leads to decrease in the food intake and increase in the energy expenditure. On the other hand, the stimulation of AGRP and neuropeptide Y, it causes increase in food intake and decrease in the energy expenditure. This pathway, AGRP and NPY pathway, it acts by inhibiting the alpha MSH, and they are the antagonists of MCR3 and MCR4 receptors. Both these pathways, they are called as melanocortin pathways of the hypothalamus. One pathway is acting antagonist to the other pathway. One of the important stimulus is the plasma glucose level. Through the glucostatic theory, it stimulates increase in the plasma glucose level, or rise in the plasma glucose level, stimulates the POMC and CART neurons, thereby decreasing the food intake and increasing the energy expenditure. And conversely, or on the other hand, decrease in the plasma glucose level increases the food intake by stimulating AGRP and NPY, and decreasing the energy expenditure. The other hormones which converge on these neurons are secreted from the GI tract, like insulin, CCK, leptin from the adipose tissue, and ghrelin from the stomach. These hormones, group of hormones, together they converge on the arcuate nuclei of the hypothalamus, thereby stimulating or inhibiting the AGRP and NPY neurons and POMC and CART neurons. We can see here the insulin and leptin and CCK, it stimulates the anorexigenic neurons like POMC and CART, and simultaneously it inhibits the leptin receptors, AGRP, through leptin receptors, the AGRP and NPY. And ghrelin is the hormone which is involved in stimulation of AGRP and NPY. The ghrelin stimulates the AGRP pathway, thereby increasing the food intake and decreasing the energy expenditure. And these other hormones, they stimulate POMC neurons in order to release the alpha MSH and stimulating MCR4 and MCR3 receptors, thereby decreasing the food intake by stimulating the feeding center and increasing the energy expenditure. So this pathway is very important, which acts through the hypothalamic nuclei. Not only the arcuate nuclei, but also the paraventricular nuclei and ventromedial nuclei participate here and regulate the food intake. Now, here at this level, I would like to tell you any changes in MCR4 receptors, very important, because of mutation leads to faulty signaling of these POMC neurons. And faulty signaling will lead to voracious eating of the individual, thereby increasing the food intake, and one of the very important factor of obesity, that is mutations of MCR4 receptors. There can be mutations of MCR3 receptors, MSH, alpha MSH, and POMC neurons. All these mutations lead to defective signaling of the hypothalamic nuclei, thereby leading to obesity. There are certain factors regulating the quantity of food intake, like short-term regulating factors and long-term regulating factors. Now, it is a point, what turns off the eating when an individual has enough eaten? Following are the factors involved in short-term regulation of feeding. One is GI filling. When the GI filling occurs, the receptors present in the stomach, they are stretched and the signals are transmitted not only from the stomach but also from the duodenum. After distension, the stretch inhibitory signals by the way of vagus nerve, these signals reaches to the hypothalamic nuclei, especially the feeding center, and inhibits the feeding center. Secondly, the GI hormones like CCK, peptide YY, glucagon-like peptides. These various hormones, they have different roles. First, coming to the CCK, when it is released in response to fat reaching the duodenum, it has a direct effect on feeding center by activation of melanocortin pathways in the hypothalamus, which just now we have seen through a diagram. Peptide YY secreted from entire GIT, the release is associated with high fat diet. The role is well studied in mice, while that in humans is still unclear. Next is the glucagon-like peptides is secreted by intestine in response to the presence of food, which stimulates the insulin production from the pancreas and suppressing the appetite. Next hormone is ghrelin, which is secreted from the oxyntic cell of the stomach. Mainly, the blood levels of the ghrelin rise during fasting and falls rapidly after a meal. This increases the food intake in experimental animals or injection while in humans. The role remains still uncertain. There is a presence of oral receptors which are metering the food intake. So there is a presence of receptors in the oral cavity which meter the food intake. In addition to short-term regulations, we have long-term regulations of food intake, in which we have the glucostatic theory of hunger and feeding regulation. The cells of the ventromedial nuclei acts as glucoreceptors. These are called as glucostats, and there is a set point in these nuclei, that is sensing of glucose in the blood. If these receptors are inadequately supplied with glucose, their activity decreases and the feeding center remains unchecked and it leads to hunger. If they are supplied with sufficient glucose, it increases the activity and inhibits the feeding center, and there is a feeling of satiety. Number 2 is the feedback signals from the adipose tissue regulating the food intake. There is a feedback signals, especially from the leptin, which is released from the adipose tissue. The role of adipocytes is very important. They function as an endocrine organ because they secrete important hormone called as leptin, and they also release a variety of factors regulating metabolism. The role of the leptin is very important. The stimulation of leptin receptors causes decreased production of neuropeptide Y and agouti related peptides, which are the orexigenic substances. This neuropeptide Y is a neurotransmitter involved in increasing the appetite. So there is decreased production of neuropeptide Y and agouti related proteins. They also cause release of alpha MSH, thereby increasing the activity of increasing the signals through MCR4 receptors and MCR3 receptors, thereby decreasing the food intake and increasing the energy expenditure. The leptin also increases the energy expenditure by acting on the vasomotor center of the medulla oblongata, thereby increasing the sympathetic activity throughout. There is a decreased insulin secretion, thereby decreasing the appetite. There is a leptin resistance which is well known, and this occurs because of mutation in the genes which are coding for the leptin. Hence, the physiologists believe that obesity is associated with leptin resistance, especially the leptin R receptors and OB gene of the leptin. The exact mechanism is still not yet established. The other specific syndromes which are associated with obesity, they are, number 1 is Cushing syndrome. Now in Cushing syndrome, we can see the obesity is a centripetal obesity. The peripheral fats from the extremities, the upper and lower extremities, it is mobilized to the trunk and the abdomen, giving rise to a pot belly obesity or a lemon on a stick obesity. Now this Cushing syndrome is because of increase in the cortisol levels. Now this increase in the cortisol level, the cortisol is a steroid hormone which acts on the adipocytes. Mobilization of the fat cells occurs from the periphery to the centripetal region. Now this type of obesity, it can be ruled out by a dexamethasone suppression test. We have also seen that obesity occurs in hypothyroidism. The obesity which occurs because of hypothyroidism is due to the diminished levels of the hormones, thyroid hormones T3 and T4. Causes decrease in the basal metabolic rate, decrease in the rate of lipolysis, and mainly because of the myxedema in females in adults. The TSH measurements rules out the cause of obesity, which is primarily because of hypothyroidism. We have certain other tumors like insulinoma, overeating due to hypoglycemia and increased energy stores that ultimately leads to obesity. We have Turner syndromes and male hypogonadism, other factors which contribute to obesity. So there are multiple pathological factors which contribute to obesity, not only the faulty eating habits and the lack of exercise and some mutations of the leptin hormones or leptin receptors, there are other pathological factors which contribute to the obesity. This is all about physiological review of the obesity. We have seen the regulation of food intake, the role of the different hypothalamic nuclei in regulating the food, the role of Professor B.K. Anand and Brobeck from All India Institute of Medical Sciences, and the discovery of feeding center and satiety centers and their ultimate role. Thank you.